Hello everyone. Thank you for attending today's talk. My name is Xiaojie Li. I'm the head of marketing at DIMA Biotechnology Ltd. Today I'll talk about the process of therapeutic lead antibody discovery. What are the challenges and what is our solution? First, the outline of today's talk. I will talk about the procedures of the therapeutic antibody discovery. And after that, I will focus on the two key steps in these procedures: the preparation of functional proteins as immunogen and the antibody development platform. Here, I will talk about our own DimAb single B-cell cloning platform and the B-cell library come from this platform. After that, I would like to showcase one example with our collaborator to demonstrate how we use our platform to find a good antibody lead molecule and utilize that at the clinical stage. In the last part of my talk, I want to briefly introduce our next day plus zero risk solutions for the antibody discovery. Let's start from the discovery procedure for the therapeutic lead antibody. There are several key steps. The first and most important step is the preparation of immunogen. Because we are developing functional antibody, the quality of the protein used as immunogen is very important for the final success. For this step, we have our own ECD and DimPro platform, which are specially designed for the membrane proteins. The second step is to build a high-throughput assay platform. A good, easy implemented assay can help us find the best antibody during the screening process. The third step of the discovery is the most tedious step. In this step, we need to develop and find as many antibody molecules as possible for the downstream screening. Currently, there are multiple technologies used for the monoclonal antibody development, such as hybridoma, phage display, etc. Here at DIMA, we have our own proprietary DimAb platform. This is built on the most advanced single B cloning technology. In the last 2 years, we used this platform to develop more than 1,000 high-quality monoclonal antibodies that covers almost 180 drug targets. The final step of the discovery is to use the in vitro assay to find the best performed lead molecule for the downstream in vivo study. For today's talk, I will focus on the first and the third step of this process. Let's start from the first step, the immunogen preparation. More than 90% of therapeutic antibody targets the transmembrane proteins. According to the transmembrane part, the protein can be grouped into two categories: single transmembrane protein and a multi-pass transmembrane protein. For single transmembrane protein, the immunogen production is easy. We can just use the extracellular domain, the ECD domain, as immunogen. The ECD part can mimic the native status of the functional domain of the protein. Therefore, they can be used to produce the functional antibodies for the drug discovery. The multi-pass transmembrane protein, the preparation of this multi-pass transmembrane protein is challenging. The ECD part of this protein is relatively short and sometimes form a short loop, so it is very hard to mimic the native status if you just express the ECD part. In this case, people use whole cell immunization to solve the issue. However, there's an issue with the whole cell immunization. The cell will break and the cytosolic and nuclear protein will be released and produce a lot of background antibodies. If your target protein is not highly immunogenic, this will be an issue. DIMA developed a series of technology platforms for the multipass transmembrane protein. We call it DimPro. These include Vyepti, Exosome, MNP, Detergent, and Nanodisc, 5 different solutions. For DIMA proteins, we not only check the purity of the protein, we also do the functional test after purification. Here a list of few assays that we normally do for our purified protein. And in the next few slides, I will also show some examples of these assays and different types of proteins. First, let's take a look at the ECD proteins for the single transmembrane target. We produce our ECD proteins in the mammalian expression secretion system to ensure their native structures and activities. In addition to the SDS page for the protein purity, we also do receptor ligand binding assay if the protein has a receptor ligand counterpart. If the protein has FDA-approved drug or in clinical trial therapeutic biosimilars, we will also synthesize the biosimilar antibodies and do a binding assays to ensure they can bind to the reference antibodies. Currently, we have more than 1,000 ECD proteins on shelf, and most of them are functional validated. We also use our ECD protein to immunize rabbit to produce a lot of functional antibodies. We have over 1,000 therapeutic lead antibody molecules in stock, and they cover more than 170 drug targets. Now let's talk about our DimPro platform, which we developed specially for the multipass transmembrane proteins. The multipass transmembrane protein is really hard to produce in a purified form or functional form. To solve the problem, we develop several solutions which can be grouped into 2 categories. Number 1, the protein is produced in a native cell membrane environment, but they are not in a purified format. Number 2, the protein is produced in both native and purified format. In the first category, the protein are produced as nanoparticles. It can be used for the immunization and immunoscreening with ELISA approach. We have 3 solutions under this category: Vyepti, MNP, and Exosome. The second category, we use special solutions to make the protein in native conformation and purified form. This include detergent solutions and nanodisc approach. So we have 5 different solutions for the full-length multipass transmembrane proteins. We use all these platforms to produce many functional proteins for the druggable target, especially the targets for the tumor therapy. I will talk about each of these solutions in details in the next few slides. Let's start from the first category, the membrane nanoparticle format. We have 3 solutions under this category. Out of them, the membrane nanoparticle MNP is the technology developed internally by our R&D scientists. Here we use a simple approach to express the multipass transmembrane protein on the cell surface and break the cells, separate the membrane part to make the nanoparticles. With the target protein on the surface of the particles. This approach uses mechanic force to make the nanoparticles. In addition to our proprietary MNP solutions, we also have the well-known Vyepti and Exosome platforms. The Vyepti use virus capsid protein. When this protein expressed in the cells, they can self-assemble into a vesicles and bud it out from the cells. When they bud out, they carry out some of the membrane fractions of the cells. If your target protein expressed on that fraction, it can be secreted out with the capsid protein as a nanoparticles. This particle is like virus, but it doesn't contain any virus genetic material, so they can't infect the cells. That's why they call virus-like particles Vyepti. Exosome approach is similar as Vyepti. The membrane budded out from the cells and carry the potential target protein on the surface. Both Vyepti and Exosome has one limitation. The budding site is on the particular domain of the membrane. If your protein doesn't distribute in that domain, they won't be able to secrete it in particle. Therefore, we recommend MNP in this category because it can carry any proteins as long as the protein can go to the cell membrane. Here is the example of our MNP protein. We express human claudin 6 on the cell membrane and make MNP proteins for this target. The left side shows this MNP protein can be coated on the ELISA plate for the ELISA assay. On the right side is the flow data using claudin 6 MNP protein. The diameter of MNP particles is about 100 to 300 micrometer, so it can be used in the flow cytometry analysis. MNP is a good approach for the multi-pass transmembrane protein preparations. However, they are not pure protein. There are some cell proteins, especially the scaffold proteins, which can produce some background antibodies when you use MNP for the immunizations. So how do we produce a multi-transmembrane protein in native structure and also in purified form? Here are our two solutions in the second category. The first solution is the nanodisc without MSP. The second solution is to use the detergent to solubilize the proteins. Detergent solubilization approach is a well-established method used for many years. The purified protein can be used for crystallization and some functional assays. However, if you are doing cell-based assays, the detergent can cause an issue to the cells. So we recommend the first solution, the nanodisc without MSP. Traditional approach, people use MSP protein to build the disc. MSP form a belt on the lipid bilayer, which form a water-soluble disc around 5 to 15 nanometer in diameter. The multi-transmembrane fraction of the protein are embedded in the lipid bilayer, and it contains both extracellular and intracellular part on the disc. The disc is very close to the native format of cell membrane, but it is small enough to carry only the target protein. So therefore it can be purified by the affinity purification. Here we use the synthetic polymer to replace the MSP proteins. Advantage of this is our polymer is not as immunogenic as MSP, so it will increase the purity of the immunogen protein and the antibodies. Here is a side-by-side comparison of our polymer nanodisc versus the traditional MSP nanodisc. On the left side is DIMA's nanodisc. We purified full-length human Claudin 6 nanodisc with our platform. You can see a single band of full-length Claudin 6 protein on the SDS page, and the purified protein also show a good binding activities in the ELISA assay. On the right side is the figure I copied from a JBC paper. This is the nanodisc made from traditional MSP approach. This data shows that you can get the purified target protein, in this case is the KOR protein, but also with a large amount of MSP protein in the same sample. So when you use this MSP nanodisc for immunization, you will also produce a large amount of antibodies against this protein. We talk about how to get a good immunogens for antibody discovery. Now let's move on to the next step, which is antibody development. How you use those proteins to develop and to find a good antibodies for the target. So for the next part of the talk, I will focus on our antibody development platform, which is called DimAb single B-cell cloning platform, and the B-cell library comes from this platform. As I mentioned before, there are a few antibody development platforms that are used to produce the monoclonal antibodies, such as hybridoma, phage, yeast display, as shown in this slide. You can also humanize the other species antibodies to get human antibodies. Single B-cell technology is the most cutting-edge and state-of-art technologies. In the following slide, I will talk about our DIMA's single B-cell cloning technology and how it is different from other B-cell cloning platforms. Here is the procedure for our DimAb recombinant monoclonal antibody development. First, the animal is immunized with functional proteins. If the animals get good immune response, we will separate B cells from PBMC or spinocyte by enriched with target protein. All the B cells will be disseminated into 96 well plate. Each well will only state one B cell. We add our proprietary culture medium to the well, and the medium will support B cell to grow from single B to hundreds or 200 B cells within 1 or 2 weeks of the time, and secrete enough amount of antibody into the medium. The antibody can be validated by traditional ELISA flow cytometry or other functional assays like blocking or activation assays. After identifying the positive clones, we will clone the IgG gene directly from these B cells by PCR cloning and next gene sequencing. After clone the gene, we will express the recombinant antibody in HEC293 cell and do an in vitro assays. Because there is no hybridoma fusion and screening step, the whole procedure is short. It only takes half the time hybridoma technology requires. The success rate is high. Using the platform, we have finished almost 180 projects within 2 years. Our success rate is around 90%. DimAb platform is robust and highly efficient. In most cases, each round of screening, we only need 1 in 20 or 40 of the total isolated B cell. A large amount of B cells are stored in a liquid nitrogen embedded form for the future use. If we need more clones, we just revive these B cells from liquid nitrogen and do another round of screening. B-cell library is one of the advantages that we offer at DIMA for the antibody drug discovery, which cannot be offered by other technologies. Here is a side-by-side comparison between DimAb platform and versus hybridoma technology platform. In terms of success rate, DimAb is much higher. In our past experience with more than 200 projects, our success rate is 90%, whereas hybridoma technology, due to the instability of the hybridoma fusion and loss of B cell during the fusion process, the success rate is much lower. In terms of the timeline, DimAb technology only needs half of the time compared to the hybridoma technology. Single B-cell technology for the monoclonal antibody development is the most advanced technology. There are many ways to do this. On the right side, I list 2 different B-cell cloning strategies used by other biopharma companies. The Beacon platform uses a highly sophisticated and expensive machine like a fax sorter to sort the B cells on a glass slide. The slide contains thousands of different vials. Each vial can only hold 1 cell and several nanoliters of medium or acid solutions. The cells are cultivated in a vial for 24 hours and secrete antibodies in the medium. Analyzing the antibodies, if they are positive, you can pick up the cells into the PCR machines. Beacon platform clone the gene truly from a single B cell without amplification. The machine is highly efficient and develops the antibody in a short time. But there are 2 limitations. Number 1, the volume of the cultivation medium in each vial is very small. The vial can only hold several nanoliter volume. The antibody produced in this small amount is not enough for any traditional ELISA or FC analysis. Number 2, the PCR amplification from single cell might have a high risk of aerosol contaminations. Another platform, oil droplet platform, is very similar as Beacon platform. It uses oil to seal only 1 cell. And again, each oil droplet only contains several nanoliter of the volume. There's not enough antibodies for the traditional acids and possible aerosol contamination from the single-cell PCR reactions. Our DimAb technology platform is different. After we isolate single B cells, we seed the cells into 96 well plate. These B cells will be cultivated in 100 to 200 microliter volume with our proprietary medium. The single B cell will be amplified from 1 to 100 to 200 cells. The medium contains enough antibodies for the traditional assays like ELISA or FC. The antibody concentration is also high. In our past test, the antibody secreted from these B cells can reach up to 0.5 to 3 mg/ml, which is good enough for any type of the traditional immunoassays. Also, during PCR cloning and next-gene sequencing, instead of from 1 cell, we get gene from 100 to 200 cells. So there's less risk with aerosol contaminations, and it is much easier to clone the IgG genes. DimAb B-cell cloning technologies can be used for many species. We can clone IgG genes direct from human, mouse, or rabbit. But we choose rabbit as host for immunization and B-cell cloning because normally rabbit antibody has higher affinity and wider epitope coverage compared to mouse. The rabbit antibody affinity is 1 to 2 magnitude higher than mouse, and if you use the same immunogen, the rabbit tends to get more epitope coverage compared to the mouse platform. The reason for these two advantages is because rabbit B-cell immune repertoire is more diversified compared to mouse. Also, the body size of rabbit is much bigger than mouse. For one immunization, rabbit gets 100 times more B cells than mouse, which means you get more clones to choose for a particular target. That's why we choose rabbit as major immune host for our therapeutic antibody discovery. Currently, there are 2 FDA-approved therapeutic monoclonal antibodies developed from rabbit. On the left is the Bevil, developed by Novartis, which is approved by FDA in 2019 for the treatment of the wet AMD. On the right side is the Vyepti, developed by Lundbeck. This is the CGRP blocker to treat migraine. This is approved by FDA in 2020. One of the major concerns for using rabbit as immune host to develop therapeutic antibody is the immunogenicity risk. Last year, Lundbeck did clinical trials for more than 280 patients after treat with Vyepti. They found there are only small portion of ADA occurrence, and with very low titer, and they are transient. This won't have any effect on the drug efficacy, and also there's no safety alert for this drug. So after humanizations, rabbit monoclonal antibody can be utilized for the therapeutic drug discovery. Now I would like to showcase one example that we collaborate with a biopharma CAR-T company. We develop a good therapeutic antibodies and CAR construct and use that for the clinical trials. First, let's go through the background of the target BCMA. BCMA is a good therapeutic target for multiple myeloma patients. In the last year, FDA approved Bluebird's anti-BCMA CAR T-cell therapy. This year, FDA approved another CAR T-cell therapy from J&J. And also, Chinese FDA also approved the ASO therapy this year. For this BCMA project, we collaborate with a Chinese CAR-T company and try to develop a CAR construct for clinical trials. From the first round of screening, we identified 13 anti-BCMA antibodies. After sequence phylogenetic analysis on the CDR regions, we found heavy chain and light chain CDR regions of all the 13 clones are different. We also did epitope binding. 2 out of those 13 clones bind to a similar epitope as Bluebird antibody, while the rest of the 11 clones all have different epitopes. BCMA has a very short extracellular domain. It only contains 54 amino acids. From this short domain, we can get so many different antibody clones, which further demonstrate rabbit as an immune host can produce a highly diversified antibodies. We choose 5 clones for humanizations, constructed the CAR, and did in vitro in vivo functional assays. One clone shows much better performance compared to the reference clone made from the Bluebird CAR. Based on the initial data, our collaborator chose 1 clone and started anti-clinical trials in March this year. Here is the summary of the BCMA project. We use 4 ml whole blood from the rabbit after immunization with our active BCMA ECD protein. From the 4 ml blood, we identify 70 ELISA positive B cell clones, and 18 works for the flow cytometry application. 13 were cloned the IgG genes, 5 were humanized, and finally 1 is in INT clinical trials. We still have a large number of B cells stored in liquid nitrogen for future screening and cloning. Now we talk about the 2 key steps for the therapeutic antibody discovery and showcased one target from the antibody discovery to the INT clinical trial. In the last couple minutes, I would like to introduce our next day plus 0 risk solutions to the antibody discovery. Currently, the biopharma company use like 2 different approach for the antibody discovery. They are either developed internally by their R&D team or contract out to the CRO company. Both options have a high risk with no guarantee for success. The company has to invest a large amount of money and time to get the good lead molecules. So to help speed up this process and push more pipelines to the clinical trials, we provide a next day plus 0 risk solutions for the lead antibody discovery. Different from the CRO companies, we are biotech companies. We provide on-shelf antibody molecules for the next day delivery. This can save at least 8 months in the development time. If the pre-made molecules don't work, we can also get more molecules from our B cell libraries in the months, which also saves a lot of time from the discovery of the new molecules. So we offer on-shelf business models. Currently, we have, like I mentioned, we have thousands of the pre-made lead molecules that covering more than 180 druggable targets. So Biopharm company can license any of these molecules and do some of the downstream evaluations with no risk. Other than the DimAb therapeutic antibody molecules, we also provide 4 product solutions, including the membrane proteins, stable cell lines, lentivirus, and benchmark antibodies for all type of the preclinical needs. Thank you for listening to this talk, and if you have any question, please feel free to contact us by email or phone. In this slide, I have included our email address and website address. Thank you.